On today, cellular respiration. And that's it for the lecture topics. As far as the labs that will be on it, there's this lab that we did on amylase enzyme, where you tested the ability of the enzyme to digest starch under hot conditions and cold conditions in different pHs. Paper chromatography lab. The microscopes lab. And the two labs we're going to do this week, cell fractionation and osmosis and diffusion. It'll be next Monday, one week from today, during the laboratory period. Same format, 100 points on the exam. Start off with some multiple choice questions and then get into some short answer, fill in the blank style questions. Sound clear enough? Alrighty. Well, let's see. We'll probably finish off the respiration lecture today, and then we'll start in on the next topic, which is called signal transduction. But none of that signal transduction stuff will be on midterm number 3. All right, and completely unrelated to any of this, one of the things that I do every year is I'm a judge for the Santa Cruz Science Fair, where kids as young as kindergartners to, I think, sophomores in high school enter projects. And I judge the botany division, even though it's not really my specialty, but you know, I don't want to judge stuff I do every day, like cellular biology here or anatomy and physiology, because that's work for me. But plants I know just enough to judge a project, but not enough to be bored with it. And oh, some of these kids are so cute. Like you get these little first graders and kindergarten girls who are, well, what did they do? They cut some flowers, then they added sugar to the water to see if the flowers grew more, then added, put a copper penny that's supposed to help the plants grow and things like that. And another group, this was actually from last year's fair, was trying to say nice things to their plants versus bad things to their plants to see if that affected the plant growth. And I forgot the bad things they said, but the nice things they said to the plant was, "You are beautiful like a star." Oh, that's so sweet. And some of the older students, like the ones in high school, some of this research is really amazing. It's almost to the level where they could publish in a journal. Like one group was studying sudden oak death, which is a parasite that's killing off some of our native California oak trees. And they were trying to see if they could correlate soil moisture to the ability of the parasite to spread from one oak tree to another. Amazing stuff. One of the perks of this job is you get to do things like that. Well, let me recap where we were on cellular respiration. So this is the big picture is it's this process that cells use to recharge their ATP supply. And what they do is they take in organic molecules, almost always glucose is their favorite fuel, and by breaking down the glucose into smaller molecules, like into carbon dioxides, that releases energy, because that's an exergonic spontaneous reaction. And the energy that's released by breaking down the glucose is used to recharge the cell's ATP supply, because the ATP molecules are the direct energy source to the cell's proteins. Remember, there were two types of cellular respiration, aerobic and anaerobic. The aerobic uses oxygen to help break down the glucose, and it breaks the glucose all the way down into 6 molecules of carbon dioxide and 6 molecules of water. And That's the more efficient type. That gets the cell 36 ATPs recharged per glucose that goes through the process. Whereas the anaerobic respiration does not use any oxygen to help break down the glucose. Each glucose gets broken into 2 molecules of lactic acid, but the cell only gets 2 ATPs per glucose that go through that particular process. And then I started talking about the 4 steps of the aerobic cellular respiration, and here are the 4 steps: glycolysis, transition step, Krebs cycle, and electron transport system. Here's a nice figure from your textbook that shows them. In the glycolysis step, glucose, which is a C6 molecule, gets broken down into 2 molecules of pyruvate, which are C3 molecules. That's the only one of the 4 steps that takes place in the cytoplasm. All the other 3 steps take place inside the mitochondria organelle. And the cell gets 2 ATPs recharged out of that step, plus some high energy electron carriers called NADHs. And then the next 3 steps take place in the mitochondria. Transition step, the pyruvates go inside the mitochondria. Well, that's not exactly true. One of the pyruvates are C3 molecules. One of their carbons gets oxidized to carbon dioxide, but the remaining 2 carbon atoms from each pyruvate, which are now called an acetyl group, come inside the mitochondria, and the cell gets 2 of these high energy carriers, NADHs, from that step. And then inside the innermost compartment of the mitochondria, which is called the matrix of the mitochondria, is something called the Krebs cycle, or citric acid cycle. And there, the carbons in the acetyls get fully oxidized to carbon dioxides, and the cell gets 2 ATPs and 6 more of these electron carriers, NADHs, and 2 more of these FADH2s, which are also high energy electron carriers. So there's the first 3 out of the 4 steps. And if you look at it so far, only 4 ATPs have been made. We're supposed to get 36, so where's our remaining 32? Well, the 32 ATPs that we're supposed to get finally come in this last step, the electron transport system. Now at this point, all the original 6 atoms, carbon atoms of glucose have been fully oxidized to carbon dioxide, so we're not going to get any more directly out of oxidizing the glucose carbons. But most of their energy is stored in the high electrons that are carried, high energy electrons that are carried by the NADHs and FADH2s. And so what the electron transport system is really all about is taking all the energy from those the electrons in those NADHs and FADH2s, and cashing that energy in, turning that energy in to ATPs. That's the whole point of this, is taking the energy of high energy electrons and converting it into ATP energy. That's what the electron transport system does. Alrighty. Where is it? Well, if you look at the inner membrane, this highly folded inner membrane of mitochondria, that's where the proteins and electron carriers of the electron transport system are. Let's see, so there's my mitochondria, and this is just to show that in that inner compartment, which is sometimes called the matrix, you get a really large concentration of these high energy electron carriers, because that's where the Krebs cycle operates, and the Krebs cycle generates a whole bunch of those. So those NADHs and FADH2s drift around inside the matrix in the inner compartment there, and eventually they bump into the electron transport system, which is in that inner membrane. Let's see that guy. There we go. Now here I'm going to show the inner and the outer membrane of the mitochondria as their phospholipid, in their phospholipid shape. All right, so here is the electron transport system of the mitochondria. Now, of course, we saw an electron transport system inside the chloroplast, the thylakoid membrane of the chloroplast. I guess that's a similarity. Remember, the thylakoid membrane of the chloroplast is highly folded and has an electron transport system. Well, the inner membrane of mitochondria is also highly folded and has an electron transport system. Notice that parts of the electron transport system look like these little tube-shaped things. Those are cytochrome proteins, which we also saw as part of the electron transport system when we were talking about the chloroplast. Remember, cytochrome proteins look like this. They are proteins that you find inside cell membranes, and part of the definition of being a cytochrome protein is it's a protein that has what's called a heme group. This is a coenzyme that helps the cytochrome proteins carry high energy electrons. And we have a protein in our red blood cells called hemoglobin that also has a heme group, but it uses its heme group to carry oxygen, whereas in the cytochrome proteins it's carrying high energy electrons. Anyway, most of the electron carriers in the mitochondrial electron transport system are cytochrome proteins. There, there, and there. The only one that's not a cytochrome protein is this little thing right there. That's called ubiquinone. It's a small non-protein. It's a small hydrophobic molecule. And its function is essentially to shuttle electrons between this first cytochrome protein and this second cytochrome protein. And actually, I should be calling them cytochrome protein complexes because each one is actually made up of a cluster of cytochrome proteins. Now, you might remember from the photosynthesis lecture what the cytochrome protein in the photosynthesis electron transport system was doing was using the electron's energy to pump some hydrogen ions, to make a hydrogen ion gradient. We're going to see the exact same thing there. That's what all three of these cytochrome complexes is going to do. All right, well, let's watch it in action and see it make ATP. All right, so inside the matrix, you have these high energy electron carriers like NADH and FADH2. Let's follow an NADH first. What happens is the NADH comes along and it delivers its two high energy electrons to the first cytochrome complex, this one right here. Oh, okay, so that's ubiquinone. Here comes the NADH. Gives its two high energy electrons, and the first cytochrome protein complex is going to use some of their energy to pump a hydrogen ion from the inner compartment, from the matrix, to the outer compartment right there. Here it goes, like that. All right, so well and good. We're on our way to building up a hydrogen ion concentration gradient with a high hydrogen ion concentration here. And of course, every time we remove a hydrogen ion from here, we're lowering the hydrogen ion concentration there. So it's contributing to the hydrogen ion concentration gradient. Well, now what happens next is ubiquinone is going to grab those two high energy electrons. They still have some energy left in them, and shuttle them to cytochrome complex number 2 right there, which is also going to pump a hydrogen ion. Fetches them. Got two high energy electrons for you. And so then pump number 2, you can think of these as pumps for hydrogen ions. Grabs another hydrogen ion, like that. And then there's still some energy left in those electrons. And so now they're going to be shuttled by this protein here, which is actually a cytochrome protein also, and delivered to this cytochrome complex pump. And we're going to pump a third hydrogen ion. And here it goes. Like that. So, now that we've got this hydrogen ion concentration gradient established, formed by the high energy electrons being used to pump hydrogen ions, now it's time to start cashing them into ATP. You might recognize this protein. We saw it in photosynthesis also. It's called ATP synthase. And remember what it does. When hydrogen ions barrel through it as they diffuse back down their concentration gradient, some of their kinetic energy is used to link together an adenosine diphosphate to an inorganic phosphate, making an ATP. And so here they go. 1, 2, 3. And so you get, the cell gets 3 ATPs per every NADH that goes through this electron transport system, because each NADH can run 3 of these hydrogen ion pumps. Sound right so far? All right, now let's focus in on the hydrogen, let's focus in on the electrons for just a moment. By the time the electrons have been used by this third hydrogen ion pump cytochrome complex, they've been pretty much drained of energy, so you can't get any more hydrogen ions pumped out of them. But we got to get rid of them somehow. They can't just, this third cytochrome hydrogen ion pump can't just keep collecting electrons forever and forever and forever. They've got to go somewhere. And this is where